Hello everyone, my name is Dalton Jenkins and I'm going to be giving a lecture I've prepared on chapter 44 of our textbook titled Osmoregulation and Excretion. Let's talk about osmoregulation. Osmoregulation refers to the homeostatic processes by which an animal regulates the amount of water and solutes it takes in and expels to keep levels within a specific range that allows their bodies to function normally. Osmolarity is the unit by which solute concentrations are measured and refers explicitly to the number of moles of solute per every liter of solution. Additionally, when two solutions have identical levels of osmolarity, they are said to be isoosmotic and will exchange molecules at an equal rate with no net flow when separated by a membrane. When osmolarity values are not equal, the solution with the greater concentration of solutes is said to be hyperosmotic. The other is said to be hypoosmotic, and water will flow from the hypoosmotic solution to the hyperosmotic solution. Let's talk about some osmoregulatory challenges and mechanisms. Osmoconformation refers to the osmoregulatory mechanism in which an animal regulates its concentrations of water and solutes by staying in an isoosmotic state with regard to its environment. Thus, the animal has no tendency to gain or lose water, and there is no net flow with the environment. The term osmoregulators refers to animals who maintain their ideal balance of water and solute concentrations in spite of external conditions, by expelling water when the environment is hypoosmotic and taking in water when the environment is hyperosmotic. Let's talk about marine animals specifically. Most marine animals are osmoconformers whose inner concentrations of water and solutes match that of their environments, so there is no net flow. Bony fishes constantly lose water to their environments, so they must consume a large amount of seawater and expel excess salts through their gills and kidneys. Moving on to freshwater animals. Freshwater animals face the issue of taking in too much water because their fluids are hyperosmotic. Freshwater animals do not drink much water. They expel large amounts of excess water through very dilute urine and replenish their stores of salts through eating and through their gills. And finally, we'll talk about land animals. Dehydration is even more of an issue for animals that live outside of aqueous environments. Some adaptations that protect terrestrial animals from desiccation are as follows. Body coverings such as skin, shells, and exoskeletons. Some desert animals have a behavioral adaptation. They are nocturnal, which reduces water loss through evaporation because temperatures are lower and humidity is higher at night. Land animals lose water through their excretory processes and through evaporation at gas exchange sites, but they gain water through eating, drinking, and metabolic processes that produce water within the body. Let's talk about the energetics of osmoregulation. Osmoregulators must expend energy through active transport to keep their water and solute concentrations within the narrow margins needed for proper bodily function. Greater differences in osmolarity between an animal's external and internal environments will require the use of more energy and vice versa. Finally, on this slide, we can talk about transport epithelia and its role in osmoregulation. So the end goal of osmoregulation is the maintenance of optimal concentrations of water and solutes within individual cells. But this is usually handled indirectly by maintaining a balance in the fluids that surround the cells. Transport epithelia are epithelial tissues that are typically arranged in tubular networks that are specialized in the transportation of specific solutes in specific directions. Here I have this diagram. It depicts a cell that is hyperosmotic to its surroundings with a net flow of water into the cell. This cell is isoosmotic to its surroundings. There's no net water movement. And this cell is hypoosmotic to its surroundings, so there is a net flow of water out of the cell. If too much water flows into a cell, it will burst or lyse, and if too much water flows out, it will shrivel. Moving on, let's talk about different kinds of nitrogenous wastes. The first form of nitrogenous waste that we'll talk about is ammonia. Ammonia is a toxic byproduct of metabolic processes that break down nitrogenous molecules that must be excreted by the body. Furthermore, many animals that live in aqueous environments are able to secrete ammonia directly because of their access to large amounts of water to dilute the highly toxic waste product, while animals without access to sufficient water must excrete ammonia in other ways. It must be diluted. The next form of nitrogenous waste is urea. Urea is the primary nitrogenous waste product of terrestrial and marine animals, and it is produced within their livers by combining ammonia with carbon dioxide. Finally, we can move on and talk about uric acid. Uric acid is the nitrogenous waste product that is typically excreted by small terrestrial animals such as insects, snails, and some reptiles. It is able to be excreted with minimal water loss in the form of a paste. However, the process of turning ammonia into uric acid is extremely metabolically taxing. Direct excretion of ammonia requires the most water, but the least amount of energy. The excretion of urea requires less water than secretion of ammonia, but more energy. And uric acid requires the least amount of water, but the most amount of energy. I have a diagram here that depicts the variations in forms of nitrogenous waste among animal species. First, there's a breakdown of nitrogen-containing macromolecules. There's a removal of the nitrogen-containing amino group, and then there is a conversion to the specific kind of nitrogenous waste that each kind of animal excretes. Moving on to diversity in excretory systems. We'll start off talking about the protonephridia. Protonephridia are tubular networks that have ciliated flame bulbs. They are structures found in flatworms and other simple animals. Each flame bulb has a cluster of cilia that draw water and solutes out of the body fluids and into the network of tubules, of which carries the filtrate to external openings where it is excreted. The metanephridia, they are excretory organs found in pairs in the columns of each segment of segmented worms and annelids that facilitate the excretion of waste products and serve to balance water uptake from their damp environments. Next, we have Malpighian tubules, which are excretory organs commonly found in terrestrial arthropods such as insects. These transport solutes and waste products from the hemolymph into the digestive tract, which are then followed by water via osmosis. Furthermore, from the digestive tract, water and many solutes are reabsorbed while nitrogenous wastes are excreted as uric acid paste along with feces. And finally, we have kidneys. Kidneys are compact, usually unsegmented excretory organs, commonly found in the excretory and osmoregulatory systems of vertebrate animals. They consist of tubule networks that carry nitrogenous waste products out of the vertebrate's body. Let's talk about the stepwise processing of blood filtrate in the nephron. From blood filtrate to urine, fluid exits the bloodstreams and enters the Bowman's capsule of the kidney, where it is converted into filtrate. Reabsorption of water, ions, and nutrients occurs in the proximal tubule of the human kidney. Salt is transported out of the filtrate and back into the body's interstitial fluids, where it is followed by water, which significantly reduces the volume of filtrate. After filtrate has exited the proximal tubule, it finds itself in the descending limb of the loop of Henle, where its solute concentration is increased and its volume is reduced even more through the loss of water to transport channels formed by aquaporins. From the descending limb of the loop of Henle, filtrate enters the ascending limb of the loop of Henle, where the membrane of the transport epithelium is impermeable to water. The ascending limb of the loop of Henle is divided into a thin segment and a thick segment. In the thin segment, salt diffuses out of the filtrate and into the body's interstitial fluids. And in this thick segment, salt loss continues through active transport rather than passive transport, which causes the filtrate to become increasingly dilute. The next stop for the filtrate is the distal tubule, inside which solute concentrations are regulated through the control of positively charged potassium ions being secreted into the filtrate and salts being reabsorbed from the filtrate. The pH of bodily fluids is also regulated in the distal tubule through the secretion of positively charged ions and the absorption of negatively charged bicarbonate. Filtrate finally makes its way to the collecting duct, where it is converted into urine. When the body is conserving water, urine becomes more and more concentrated as it moves through the collecting duct due to water reabsorption. When the body needs to rid itself of excess water, the membrane of the transport epithelium lacks aquaporin channels and does not allow water to diffuse out of the filtrate. Thus, the urine produced is very dilute. We can talk about solute gradients and water conservation. The mammalian kidney is effective at conserving water but must expend large amounts of energy to produce hyperosmotic urine by facilitating the movement of water and solutes against concentration gradients through active transport. Talk about how urine is concentrated in the mammalian kidney. Filtrate passing into the proximal tubule from Bowman's capsule has a large volume and about the same osmolarity as blood. The filtrate experiences a loss of water and salt at the same time, so its volume decreases while its osmolarity remains about the same. Filtrate then passes into the descending limb of the loop of Henle, where it becomes more and more concentrated. Once filtrate enters the ascending limb of the loop of Henle, salt begins to exit the filtrate and enter the interstitial fluids of the renal medulla, which helps maintain the osmolarity of these fluids. In the loop of Henle, solutes are exchanged between two oppositely flowing fluids through a process known as countercurrent exchange. Additionally, this specific countercurrent exchange system present in the loop of Henle employs active transport to control the movement of water and solutes against concentration gradients, which makes the system a countercurrent multiplier system. The vasa recta also employs a countercurrent exchange system. As the descending vessel transports blood to the inner medulla, it loses water and gains salt, while the upper vessel carries blood back to the cortex. The opposite is true. The kidney, mainly the thick segment of the ascending limb of the loop of Henle, must expend a large amount of energy to facilitate the active transport that maintains the steep osmotic gradient between the medulla and the cortex. Because of the active transport that takes place in the thick segment of the ascending limb of the loop of Henle, the filtrate is hypoosmotic to body fluids as it makes its way into the distal tubule. From the distal tubule, the filtrate flows towards the collecting duct, where it becomes more and more concentrated because of the loss of water to the increasingly hyperosmotic body fluids it makes contact with. And finally here, we can talk about adaptations of the vertebrate kidney to diverse environments. We'll start off with mammals. And mammals that often face the issue of dehydration typically have many juxtamedullary nephrons with very long loops of Henle because these adaptations allow them to produce highly concentrated urine and conserve as much water as possible. Mammals that live in aquatic habitats do not often face the issue of dehydration typically have cortical nephrons that do not allow for the production of very concentrated urine. Next, we'll talk about birds and some other reptiles. Most birds have kidneys that have juxtamedullary nephrons and loops of Henle that are not as long as those found in mammals, so they cannot concentrate urine to the same level as mammals. However, birds conserve water by excreting uric acid as their primary form of nitrogenous waste. Other reptiles typically have kidneys with cortical nephrons. They excrete uric acid as their primary form of nitrogenous waste, and they reabsorb water from their waste products through the membranes in their cloacas. Freshwater fishes have kidneys with many cortical nephrons, and they produce very dilute urine at a very high rate. Furthermore, they maintain solute concentrations by ion reabsorption that takes place within the distal tubules. And amphibian kidneys function very similar to those of freshwater fishes while they are in an aquatic environment. However, on land, amphibians conserve water by reabsorbing it across the epithelium of the urinary bladder. And finally, marine bony fishes. They produce much less filtrate and have kidneys with far fewer nephrons than those of freshwater fishes. Instead, the main function of the kidneys of marine bony fishes is to rid the body of divalent ions, which are ions who have a charge of +2 or -2. Here I have a diagram that depicts the flow of filtrate through the nephron. It starts off in the proximal tubule. Passive and active transport are both going on here. Then we move into the descending limb of the loop of Henle, where water is lost through passive transport. And we make our way to the ascending limb of the loop of Henle, where the thin segment of the ascending limb, where salt is lost through passive transport. Then in the thick segment of the ascending limb, salt is still lost, but it's through active transport. Then we make our way to the distal tubule, and both active and passive transport are going on. Salt, water, bicarbonate are leaving the filtrate, and potassium ions and hydrogen ions are entering the filtrate. Then we move down to the collecting duct, where salt is still being lost through active transport, and through passive transport, urea and H2O, water, are diffusing out of the filtrate. Finally, we can talk about some hormonal circuits and the homeostatic regulation of the kidney. First, we'll talk about antidiuretic hormone, or ADH. Vasopressin, or ADH, is a hormone present in the kidney that responds to high blood osmolarity and results in the formation of extra aquaporin channels, thus increasing the amount of water reclaimed from filtrate. Next, we have the renin-angiotensin-aldosterone system, or RAAS, which is a circuit present in the human endocrine system that prevents dehydration by employing a specialized tissue known as the juxtaglomerular apparatus, or JGA, which produces an enzyme called renin that cleaves the protein known as angiotensin to create a peptide known as angiotensin II. Furthermore, angiotensin II raises blood pressure through vasoconstriction and stimulates the release of the hormone aldosterone, which stimulates an increased rate of water and ion reabsorption in the kidneys. And finally, we can talk about atrial natriuretic peptide, or ANP, which is a hormone that responds to high blood pressure and acts as a counter to RAAS by inhibiting the release of renin, inhibiting the reabsorption of sodium chloride, and inhibiting the release of aldosterone. ADH, ANP, and RAAS work together to balance the osmolarity of body fluids. And that is all I have for today. Thank you so much for listening and have a great day.